All right. You all took Ellen's crash course, I imagine, last week, and if you were not feeling confident about neurophysiology or anatomy a little, you must be now doing a whole lot better. That's my expectation. But if you didn't get a chance to attend, the recording is also available, so you could also watch that and kind of see where. And if you have questions about it, of course, you could ask the TAs or in the class or something. But that was meant to give you the background in order to understand the material in the course. Sorry. Any questions? Any questions about the syllabus, the course? Yeah.
Is the paper, the first assignment, is there just going to be one paper that's available to do the assignment on? Because there's one in the syllabus right now.
Yeah, I'm going to add another one to, so thanks for bringing that up. So I did put, so let's go to the syllabus, talk about it. So I put a comment here. I'm going to add another paper here, but this paper is available if that's something you're interested in to do the assignment on. And I did add this Canvas. I did add some stuff. So on the Canvas page, you could go to modules, and I go to student view so you can see this. We go to modules, and here you see that I have something called assignments and paper templates. This is a paper that was assigned last year. And if you're interested, you could take a look at it. It's a nice paper. And then I also have included 2 sample assignments from students that we thought were good. This is from last year. And you'll see, and it's, again, it's to give you an idea of what we expect, and we don't expect something perfect and so clean and whatnot. It's something very reasonable. And if you read the paper and you read these, and you'll exactly know what we're looking for. And there is a template here that you'll use to format your response. Basically, this is a Word document. You could download it. You could fill it in with your responses. But the paper is not this paper that I have here. This is just a sample paper for you to match it to the response that the students wrote in order for you to understand what the expectation is. Here also there are other relevant stuff, the stuff that we have in the first module, the lecture slides, the tutorials. We'll add a couple of more later on, and then the textbook. But if you like that paper, you could just start working on it. I'll add the other one in the next few days. I'll add another paper here, and then you'll have your choice between the two. Is that good? Other questions? Great. So today, we're going to talk about some neural network stuff and some different neuroscience techniques. So where did we leave off last week? We left off at this slide where we talked about how different neurons can communicate. And we talked about excitatory connections and inhibitory connections and how they could, so how one presynaptic neuron can impact another postsynaptic neuron. So you're all familiar with that terminology now when I say a presynaptic neuron, a postsynaptic neuron, right? The direction of the effect is always from presynaptic neuron to the postsynaptic cell. And you know that relationship could be excitatory or inhibitory, right? So what makes a presynaptic neuron excitatory versus inhibitory? So Yeah, so what properties, what's different between a excitatory versus inhibitory presynaptic cell? So that's the impact it has on the postsynaptic cell. What is it? No, a postsynaptic cell, whether excitatory or inhibitory, could have a high firing rate, right? Well,
But is it a difference between like hyperpolarization or?
yeah. So what you're talking about is the impact that the presynaptic cell has on a postsynaptic cell. But the presynaptic cell could be an excitatory cell or inhibitory cell. What yes. Yeah, so it is based on what kind of neurotransmitter this presynaptic cell releases. If it releases excitatory neurotransmitters, it is an excitatory presynaptic cell. If it releases inhibitory, then it's an inhibitory presynaptic cell. So what is the effect that has on the postsynaptic cell? If it's inhibitory, what is the effect it has on the postsynaptic cell? So what does it mean to inhibit a cell? What is the observable behavior of a cell that you say, okay, it's inhibited? What is an observable behavior? Okay, so what is the behavior that we could observe from the cell? It's not firing. So the behavior that we could observe from a cell, it's firing rate. That's the behavior that we could observe from a cell. That's what I was in the video I was showing you. That's what we're listening to from the cell. That we're listening to how many times the cell is firing. That's the behavior. And you could tell if a cell is being excited or if a cell is being inhibited based on that behavior. If it's being inhibited, what does that mean? What kind of input is the cell getting? What does it mean for its presynaptic cells? What can you, if a cell is not firing that much, what can you tell about its presynaptic neurons? The presynaptic cells? A cell would not be able to regulate its own presynaptic messaging, right? A cell can fire. A cell, a neuron, what it can do, it could either have an action potential or not. That means that it could communicate the signal to its postsynaptic cell or not, right? So imagine you have a cell, it has presynaptic stuff coming into it, and it passes on information. In here, as I'm showing in this figure, when a cell is not firing, what does it mean? Hmm? It's inhibited. But what does it mean for the type of input that it's getting? It means that it's getting a lot of inhibitory input and very little excitatory input, right? So if something, so what does it mean in a car when a car is coming to a stop? What does that mean? It means that you're pressing the brake pedal and just not, picked your foot up off the gas, right? So every cell is getting a ton of, so you could imagine every every synapse that comes to this cell, it's either a gas pedal or a brake pedal. Does that make sense? And a cell is accumulating all this. So imagine a car with 500 gas pedals and 500 brake pedals. And a cell, if it's going really fast, it means that, well, all these gas pedals or some of them are being pressed down. And if it's slowing down, it means that what? It could mean two things. What does that mean when a cell slows, when it slows down? Yeah, so it's getting more inhibitory input than it's getting excitatory input. So every cell, as I said, a car with a lot of gas and brake pedals, if the brakes are stronger than the gas, it's going to inhibit the cell. if the gas is stronger than the brick, then it's going to fire. Like it's going to increase the firing rate of the cell. Are we all clear on this? This is the takeaway, at least from last week. This week, we're going to talk about other stuff. So, yes. What's that? I am recording. Am I not? What's that?
I was just making sure. Sorry.
Yeah, I think there's this little red dot in here means it's recording. Yeah. Thanks for checking. Sure, yeah. There are a lot of excitatory and inhibitory neurotransmitters. We didn't get into them in this course, but some of them, yeah. The two examples you said are correct. Right? Glutamate or something like that is excitatory, GABA is inhibitory. For the purpose of this course, we really don't get into them. But what I just described, right, is an impact of, let's say, 2 or 3 or a bunch of cells on each other. But as I said, in the brain, we have, tens of billions of cells, and they could communicate with one another. So you could imagine the complexity of how they communicate and how kind of behavior you could see even in 2 cells. So inspired by that, people have built what we call artificial neural networks. And these would be models that actually try to describe the behavior of a neural network. But they are very abstracted, right? They don't worry about all the detailed neurophysiology of a single cell, but they just abstract everything to the firing rate and the impact that cells have on another. So, but you all know about artificial neural networks now, right? We've all seen them around. What is a good example of an artificial neural network? What is an example AI model? ChatGPT, right? ChatGPT is a big neural network. A lot of the stuff that like Google search uses neural networks, or a lot of image recognition, the way, for example, Google Photos organizes photos of faces and houses and places and whatnot, uses neural networks. So we've seen Google Translate uses neural networks, right? All of these systems that you see in everyday life, these are neural networks. These are massive neural networks, but we're going to look at a very simple one, just for you to have an idea of what these things are and what does it mean to compute with this set of neurons. So these are the inputs, imagine, of this hypothesized very small toy neural network. And these are the outputs. So imagine each of these is a neuron. So we have 8 neurons here. Which ones are the presynaptic neurons? Yeah, the inputs, these are the presynaptic neurons. And these are the postsynaptic neurons. So the direction of effect is from bottom to top. So neurons at the bottom are impacting the activity of neurons on the top. Very simple so far. If it's a 0, it means that this connection doesn't exist, that this neuron is not connected to that neuron. If it's negative, it means that it's an inhibitory connection. If it's positive, it means that it's an excitatory connection. Okay? And the values that we have here, these are basically the connection strength. How much can one neuron impact another neuron? So in a very, again, it's a very abstract way, these connections are the way one cell communicates with another cell. It's the way that we talked about. These are the synaptic strength between 2 neurons. And we call them connection weights. And the current understanding is that all our memories, all our knowledge, everything that we know is stored in these connection weights, right? 2 cells, or, there are trillions of connections in the brain, And those connections are where our knowledge is stored. And it's the same for a neural network. So let's say you have a pattern of activity here. When it's positive, it means that the firing rate is above the baseline. When it's negative, it means that the firing is below the baseline. It means, so these cells are getting input from other cells, right? So these are postsynaptic cell for another something else. So this is a neural network and there are many, many layers, right? Something like, say, an object recognition network may have up to 200 layers. Here we're looking at one. But imagine this is the current state of these neurons, that these two are firing above their baseline and these two are firing below their baseline. Now we could, and again, imagine that this pattern means something. Now we could look at, well, if this is an abstract neural network, can we think about, okay, what would be the firing rate of neuron output one, right? We could calculate that. How? Well, you're saying neuron input one has no connection, so it doesn't matter what its activity is. Neuron connection two has 0, so this is also has no impact on the output one. You could say neural input 3 has -1, and you multiply it by the connection weight, and this is an abstraction, as I said. You could say the impact that one cell has, there are 3 factors that goes in the connection, in the impact one cell has to another cell. One is, what is the activity of this cell? In this case, it's below baseline. 2, what is the connection weight of this cell to the postsynaptic cell? 3, what is the strength of that connection? Do you see all three factors here? So what we have here is that you basically multiply that what we call, let's say, firing rate by the connection weight, and then you put it, this becomes the impact of input 3 on output 1, and the impact of input 4 is similar. 1 times this -0.5 becomes -0.5. Then you add those up. And again, I'm talking about a very abstract neural network. I'll finish this and I take questions. So what this means, that the neuron output 1 is inhibited, that its firing going to be below its baseline. So why would neuron output 1 be inhibited, right? From what I said earlier, why would output 1 be inhibited based on these 2 inputs? Well, because when a presynaptic neuron was active above a baseline, right? You're not seeing my thing. I'm sorry. I have to find a better way to communicate this. When the input 4, so input 4 is the connection of input 4 to output 1 inhibitory or excitatory? Let me start. It's inhibitory. Is the activity of input 4 above baseline or below baseline? It's above baseline because it's 1. So if a connection between, if this neuron has an inhibitory impact on neuron output 1 and it's firing above a baseline, what is the net effect for the output 1? It's inhibitory. Does that make sense to everyone? That's the takeaway I want. No?
But what doesn't make sense is when you said output 1 was not inventory.
No. I'm saying, so the connection from input 4 to output 1 is inhibitory because it's -0.5.
Right.
That's an inhibitory connection. The input 4 is firing above a baseline. So if input 4 is firing above a baseline and it has an inhibitory impact on output 1, so the net effect on output 1 is what? Inhibitory.
How do we know that input 4 is above baseline? Is there a 1 underneath?
Because yes, because it's positive at the bottom.
Okay, and what do the bottom numbers represent?
Bottom numbers represent the state of the input neurons.
Got it.
So I call this an abstraction. Again, this is very abstract. I just wanted you to have some small familiarity with what a neural network is. So it's a very abstract. I could probably better duplicate my screen so you could see these things better. Let me see. No, it's coming from outside. Right? Or no?
I'm just gonna go check it out. It sounds like a flare. I'll go check it out.
Oh, check it.
You have questions?
Yes. Wait a second. Let me see how I best can do this. Yes, question. Yeah.
So just thinking about this, if input 4 was -1 and it was firing on output 1 as -1, would it then be excitatory?
Yes. What does that mean? In my car example, what does that mean?
It means that
It means
going down a hill and you're taking your foot off the brake.
you're taking your foot off the brake. So if, because this is negative, it means that it's a brake, right? At least from the polarity of these connections, you could tell if it's a brake or a gas. So if this is a brake and this is negative, it means that you're taking your foot off the brake. And the net effect on output 1 is positive.
I don't know if you're like pointing at something.
No, I know. I'm trying to share my screen, and this is a, but for some reason on this keyboard, I don't see the. I have to. Hmm. Was it? Oh.
I mean, see your mouse now if you don't want to make it completely full screen.
Yeah, I'd rather make it full screen, but it says duplicate. So why wouldn't it? Oh no, I just moved it over. Don't worry about it. But it's okay. I'll make it work. So exactly. So here, let's say for another output, let's say you have output 4, right? So you could see that, okay, output 4. Let me get one that has excitatory. Okay, so output 3. Output 3 has this 0 connection, right? So nothing here. This is also 0, but it takes input from input 3 and input 4, right? Input 4 is an excitatory connection to output 3, and it's above baseline. So what is the net effect on output 3? Excitatory. So you just say 1 times 0.5, it becomes 0.5 for output 3. Here, it's -1 times -0.5 is 0.5, and that's also excitatory. So the output 3 would be 1. Very simple. You want to multiply the state of a neuron times its connection, and then that gives you what is the impact of this specific cell, presynaptic cell, on the postsynaptic cell. Does that make sense? Again, I think the metaphor of gas and pedals kind of work here. So input 3 is like a brake to output 3. It's an inhibitory connection to output 3. So if it is below its baseline, meaning that you're not pressing the brake button that hard, it's going to make output 3 go faster. I can't make it simpler than that. So if there is a misunderstanding, we have to. Input 4, it's a gas pedal because it's a positive connection to output 3. So if it's positive, if it's really strong, then the net effect on output 3 is positive. And this is also because you're picking off of a brake pedal, it's also positive. So output 3 would be positive 1.
And then you add all of them.
Yes. So every cell, every neuron is collecting all these inputs from all its connections. And so you basically calculate what is the effect of this presynaptic cell on that postsynaptic cell, what is the effect of this presynaptic cell on that postsynaptic cell, and then you add them up. And the effect of a presynaptic cell on a postsynaptic cell depends on 3 things, basically. What are the 3 things that I mentioned?
The activity of the cell, the connection weight, and the strength of the connection.
Exactly, right? So what matters if this cell is connected, if it's impacting this cell, it matters what type of connection they have. Is it excitatory? Is it inhibitory? How strong is this connection? It could be a really strong connection. It could be a really weak connection. And also, how active is this cell? If this cell is not active, it's not going to, if this cell is at baseline, it's not going to change the activity of the postsynaptic cell. If it's above or below its baseline, that it could really impact the postsynaptic cell.
How quickly does the communication go from postsynaptic cells?
It's like in the order of maybe 10 milliseconds. Depends on how far they are from each other, but it's fast.
And they're all like connected to each other. So this is happening like cross interdependent kind of network.
Yeah. So a lot of this is happening, right? At any given moment, you have a lot of synapses that are active and going on. So it's a complex system. This is a 2 example. Something like ChatGPT has, for example, what number? So each of these connection weights is called a parameter in these models. And that's, as I said, all the knowledge in a network and the brain is supposedly stored in these connection weights. Whether a connection exists, whether it's positive or negative, how strong it is. That's where the knowledge is stored, and that's what the parameters of a given model is. So how many parameters are in like latest version of ChatGPT or a large language model? Does anyone know the order of the number of parameters in, let's say, a large language model? Take a guess. Sure, yeah, it's around 100 billion, for example, right? So you have a system. And what is ChatGPT? The ChatGPT is basically takes a sentence and tries to predict the next word, right? The core of it is that simple in terms of what you train it on. So you basically tweak these connection weights. So when you have a sentence, for example, patterns of excitatory and inhibitory activation over the input, and then you mask out the word nodes, and ChatGPT would try to output the word nodes. So that's what it is. Practically, that's what it is. It's just a massive network. Yeah.
The math equation again, I'm a little confused how you got negative.
So let's say output 2. So you answer the question. So how many cells in the input are connected to output 2?
Output 2?
Yeah.
1 and 4.
So 1 and 4, right? Is it or no?
Okay, what do you mean?
So how many of them actually have non-zero connection to output 2?
Oh, non-zero. Oh.
Sorry. So it's input 1, right?
Yeah.
Input 2, and that's it.
That's it.
Okay. So we have 2 actually input that are actually non-zero connection to output 2. So let's say input 1. Input 1 itself has inhibitory or excitatory connection to output 2. Inhibitory. And input 1 itself is above a baseline. So if it's above a baseline, it's really active and inhibiting the postsynaptic cell, what would be the effect on that? Well, it would be 1 times negative 0.5, it would be negative 0.5. It's inhibiting the cell.
Oh, okay, so you multiply
The state
this state by the, I see.
by the connection. How about input 2? Well, input 2 is excitatory to output 2, but itself is being inhibited. So it cannot excite this. Instead, it brings that down. So it becomes negative 1.5 times 0.5. It's negative 0.5. You add two negative 0.5s and they become negative 1. Very simple.
Just to make sure, okay, so the difference between connection weight and strength of that connection. So the strength of that connection is whether it's plus a positive or negative, inhibitory or
So when I use the word strength, I meant how strong it is.
okay, so
But the type of connection, I would say excitatory or inhibitory. So the terminology I use is that type of connection is excitatory or inhibitory. The strength would be how strong that value is.
a connection weight,
Yes.
like negative 5.5.
So that's what is called the connection weight or a parameter in a model.
But like, just to make sure I understand,
Sure.
connection weight is that number, whether or not it is
Positive or
positive
negative.
or negative. It will determine obviously whether it's going to be in the end excitatory or inhibitory based on the baseline of the presynaptic.
So it will based on this based on it says negative 0.5 or 0.5, you know if it's excitatory or inhibitory. But this could have been a 5, for example. Let's say this was a 5. Well, if it was a 5, you would multiply that 5 by this 1 and be like 5 plus this negative 0.1 times negative 0.5, which is sorry, I'm doing it wrong here. So negative 0.1, negative 0.5, so it's 0.5 here. But if this was actually a 5, it would multiply 5 by 1, and then this would be, let's say, 5.5. Okay? So it depends, meaning that if two cells are really connected to one another, that response would dominate the other inputs.
Car is like driving slower than it was before, than its baseline.
Yeah.
And then it releases an inhibitory neurotransmitter. So like, I don't really understand why that's taking your foot off. Or like,
Which one? Which input are we talking?
so let's say input 3, and the car is
To
going slow, and then there's
so input 3 to what?
input 3 to output 3.
Okay.
And the car is driving slow, and then there's a neurotransmitter. So in my mind, that's like they put on brake. So why would it?
Not necessarily. This is a brake. Like this is a physical brake, but that doesn't mean that you're pressing it. If it's a negative one, it means that you're not pressing it as hard.
So if let's say instead of 0.5, it was 1.5, then you would be pressing the brake.
Instead of negative one, you mean?
No, from minus 1 to minus 1.5, yes.
Oh, you mean here?
Yeah.
No. This wouldn't be pushing the brake. If it, you're saying from minus 1 to minus 0.5?
Yeah, you're saying that negative 0.5 isn't necessarily pushing the brake, but I guess I'm asking if it was negative 1.5, would it be?
Okay, so let's take two things away. I'm gonna so this value tells you whether it's a brake or a gas, let's say. If this is negative, it's a brake. Let's go with that. Let's go with that metaphor. What this can tell you is whether you're pressing the brake or picking your foot off the brake. If it's negative 1, let's say you're picking your foot off the brake. Does that make sense? What's the problem then? What's the confusion?
I guess I'm just trying to understand how one the analogy of driving a car is to this.
So the analogy is, I'll go through it and tell me where it's confusing. This is a brake, right? Input 3 to output 3 is a brake. And if this was 0, it means that this neuron is at a baseline activity. And it does have no impact on the postsynaptic cell. If this was 1, it means that it's going above its baseline activity. And because this is a break, it would slow that down. Now, if it's -1, it means that it itself has gone quiet. If it has gone quiet, it cannot release even its baseline inhibitory neurotransmitter. So it in turn, this would go up. Make sense?
So the state numbers on the bottom, a positive one would be putting foot on a pedal, even if it was gas or brake.
Yeah.
Okay.
So this here, it just says whether this activity of this cell is above or below its baseline. If it's a break, it still has to, if it has a break impact on the other cell, it still has to go above 0 to be you actually pressing it. The confusion is that in that quadrant, right? The quadrant is that if it's inhibitory, if the connection is inhibitory, but this cell itself, this presynaptic cell, is inhibited, then it cannot inhibit. You see what that means? If a presynaptic cell is inhibited, but it was inhibitory to a postsynaptic cell, then it cannot inhibit the postsynaptic cell because it's not active. So in turn, it has a positive effect on the postsynaptic cell. So that's what you see with negative times negative. Work through it yourself. You'll get a hang of it. But the thing is, metaphorically, right? These patterns mean something in the brain. If a certain set of neurons is active versus certain other set of neurons is active, it means different things. If you hear the word duck or if you see a duck's face, you may have this pattern of activity in a host of cells in a certain visual area in your brain. If you see another set of activity, then it means that you're perceiving something else, right? These activities mean something in the brain and probably in this network. So you could say, well, if I have one input vector, I would have a certain type of output vector. If I change the input vector, I could get another output vector. So this is what we have in our brain. When we hear a certain kind of word, we perceive something. When we hear another kind of word, we perceive something else. It means that the fact that you're mapping from a certain, let's say I would say, imagine a duck. Well, you imagine a duck. If I say imagine a penguin, you imagine a penguin, right? What happened there? Well, you went from that word that you heard to an image of the animal. And that happens over many layers of a network, but it's the same network. It's a shared network, right? So it's one network. You give it one input, which is a certain pattern of sound that you hear. You hear another input, which is a certain other pattern of sound that you hear. They feed to your network, and at the end, you have another patterns at the end versus the other. It's an abstraction, but it is to show you that a certain pattern can emerge from the same network. And the reason it does is because all your knowledge, all you know about penguins and ducks are stored within the weights of these networks. Yeah.
I don't know much about AI, but is it fair to say that that's what AI is?
Sure.
I think I'm just.
So this is, yeah. So this is a small network with like 16 parameters. But imagine this could be a huge network with billions of parameters. And you give it an input and you get an output, right? So it's at the core of it is this. It's units that have certain states. They, using these parameters or connection weights, they become new states in the next layer, next layer, next layer, and over 150, 200 layers, they become something else. So a word duck, you could translate it to an image, or you could translate it from one language to another language. Yes. No, I guess they get stronger when you get older. Like as you learn more, I think that the core of it is like developmentally they become stronger and more more set as you get older. Obviously, there are a lot of learning later in life, but not as much. Like it becomes harder to learn a language or something.
The input baseline, that can that ever change, and it's also dependent on
Yeah, so imagine that the input will get there, but imagine the input is the photoreceptors in your eye. So if I see a pattern that is brown, the photoreceptor is going to respond differently than a pattern that is red. So these inputs are, let's say, the firing rate of those photoreceptors, and they respond differently to different visual input. But then these different inputs feed to the same network, and then at the end of it, you have a perception of a red or perception of brown. That's what that means. Sure. I think it depends on what you mean by that. So there are a lot of, it's hard to say how much of connectivity, let's say, is coded in the genes. It's probably not much. It's a structure that is coded. And then with learning, you kind of fill in all the little connection bits. It's a learning based thing. But some people argue maybe in more primitive animals, a lot more of these connections are already, they're born with these connections. Like there are animals that could just, as soon as they're born, they're kind of adapt to their environment. They can walk around. It's not true of human babies, but it's true of a lot of animals that they know a lot of things. And the fact that they know and they can respond to visual input, they could run away, they could find food and all that. It means that a lot of these connections are pre-coded.
Does the speed of the firing changes as you grow, or is it
I don't know about that, but that's an interesting question. I don't know if there is a way of quantifying whether, but again, the speed of firing is different depending on which area of the brain you are, which type of cell you are. Some cells fire on baseline, they fire like a lot. Some cells are fairly quiet. It depends on which area of the brain it is. I think that matters more. Yes.
on this on this spatial timeline. Like I think you said last time, but also depends on if the firing is simultaneous or not. Like, didn't you say something like this? Like one postsynaptic cell receiving
Mm-hmm.
excitatory or inhibitory signals from a presynaptic.
Go ahead.
Like the timeline matters too, right? Like it's let's say.
So suppose meaning that whether they all come at the same time or not? Yeah.
Or not.
Yeah. I think for a cell to actually, so a lot of cells, you could imagine that they detect this coincidence. If a lot of, for one cell, a lot of things have to come together for it to actually fire, right? So meaning that if you want to recognize a face of a person, you're detecting a lot of things about them, and then they feed through multiple layers, and then they become a certain pattern of activity. Um, but for one cell, yes, a lot of things, a lot of presynaptic stuff should be active for this cell to reach a certain threshold and actually emit an action potential. Not only that, but continually do that, right? You don't communicate with just one action potential. You actually fire for an extended period of time. So a lot of the knowledge, as I said, it's important to internalize these points. The knowledge, the memories that we have are stored, the trauma that you have are stored in these connection rates between different cells. And it's true of these networks too. But every perception that we have is basically an state of the neurons in our brain, right? So in this state or in that state, any perception that you have has to translate to a state of neural activity in your brain. And computation is basically converting one set of representation to another set of representation, to another set of representation. That's what it is. That's why we call the brain is computing this or computing that, because it's basically getting a certain kind of input through sensory inputs and then converting it to maybe a concept that you know. Like you look at a picture and you say, okay, this is a duck. How does that happen? Well, you have a visual input on your photoreceptors. It becomes something else. It becomes something else. And over many layers, it goes to a certain area. And if it's a certain pattern of activity that you knew from before, it becomes a person. What's about this is so different? So I use a computer metaphor. What about this is so different from, let's say, a computer like the laptop in front of me? Something about this is very different. Sure. Some people may argue that that action potential is like a zero, it's an all or none, but that's a fair point. But there is a much bigger difference here.
The laptop is perceiving anything. We get competing.
Maybe you are perceiving. That's fair. I'm looking for something else. No. Look at the network. What? No, this is gonna be in a computer the way it's simulated. But I'm just saying this is different from a computer. What did I say? What did I say about this network? What is the quality of this network that is it's even written on the right side, on the left side of me? Yes. What is the memory of this system? What is the memory of a laptop? It's a hard drive. It's not in the connection weights of anything, right? These are hard drives. These are the systems where you write. It's basically gates that are zeros and ones, and they store information. They're not in the connection weights of anything, right? And this is a fundamental difference between what we think, what we call one human computers, like a laptop or the brain. So it's something to think about. So this metaphor kind of works, as I said, because you could think about how going from a sensation, right? Going from the light weight hitting your eye or a pattern of sound hitting your ear, and we'll talk about this later on. But going from that to a percept, to a memory, you could think of it as this cascade of neural activity traveling in your brain, traveling across multiple layers. It's a simplification, but imagine you have sensory processing, you attend to something, right? You attend to this part of the world or that part of the world. You recognize it, and then you attend to it more, and then it becomes short-term memory, long-term memory. These are concepts that we get to, but in a network, you could imagine a simplified version of this. All right. Let's take a 10-minute break. 6:53, 7:03 we'll start.
Before people leave on break, if you haven't signed the attendance sheet,
Okay, so next we're gonna talk about single cell neurophysiology. This is the practice of basically recording from a single cell. And I showed you something last week on this, where given that a single cell voltage inside of it, it's gonna increase above a certain threshold, emit an action potential, and then it come down. And then for each action potential, it's gonna do that again. So if you put an electrode right next to that or on the membrane, you could actually record that activity going up and down. And if you could record the activity going up and down, it's really small, but we could amplify that, and then we could feed that to a speaker and then listen to it. That way you could see what a single cell is doing across a period that the experiment is going on, across different visual inputs, across different tasks, right? But a lot of early work in this area was done, as I said, with anesthetized animals. So they weren't active, they weren't doing anything, they were basically put to sleep. That way it makes a lot easier to run experiments. And if you don't want to study something related to awareness or conscious behavior, then it makes sense. So one thing to note is that in the visual areas, for example, you could really see if you flash a light on the screen, you could really see certain areas that the cell is responding to. So, and this is something that I was showing last time. This is an area, we'll get to it. So what these experimenters are doing, they have a screen, they have a light, and they could change the shape of this light and then add LED to it, make it bigger, give it different shapes. And they're just basically sweeping the screen with this light. And this is all done manually. This is way before the modern computers and stuff like that. So it's all manually, they're moving it around and they're recording from a single cell in a certain brain area that gets input directly from the eye. And we'll get to it. Later on, we'll talk about different pathways. But for now, let's just listen to this single cell. What you see is that as soon as the light goes on, you could hear the cell firing. Each of that dots that you hear in is an action potential. So that's the firing rate, how quick it is.
Are we looking at the cell somewhere on this screen?
No.
Or we can't see it?
No, it's just a single cell in the brain. No, you're looking at the screen that the animal is seeing.
Okay, got it.
Yeah. This is the screen that animal is seeing. But what these people are trying to find out is that this is specific cell. It looks like it likes light at the certain areas of the space, right? That's so we'll watch more of that later. But for now, this is basically what a single cell neurophysiology looks like. Now, the area that you saw, so as you saw, the experimenters were just playing around with this light to see which part of that the space maximally activates the cell. And again, I'm using different words to say increase firing rate, like activating, exciting, or this is something that this cell likes. All of those means that makes this cell have a higher firing rate. And that area is called a receptive field. A cell could have a receptive field, a response, but these receptive fields are only in the areas that are close to the sensory input. They're not really, when you go higher up, and we'll talk about what higher up and lower level means in this hierarchy, but that's what the receptive field is. All right. And it's an important tool really to understand what cells like. But as I said, it's not something that you could use everywhere in the brain because it has to be somewhat close to the sensory input. Like you could do that with some different patterns of light or different images or stuff like that, but you can't really do it with more complex stuff. It's mostly about things that are sensory processing, closer to perception rather than cognition. So in a way, this is another terminology that people use. If a cell, right, is active for a certain pattern of light somewhere in the visual input, or an oriented bar, or a face, or something like that, you could say that this cell is coding for that input. Like in a way, this cell, when there is that input in the world, this cell gets really active. And by its activity, it communicates something to the rest of the brain. It communicates, okay, that there is a light there, or there is a bar there, or something like that. And then some other cells can use that information and process that. And then some other cell can, so there's a cascade of processing, which starts from simple detection and perception and coding, and then it gets progressively more complex. So you start from these bars, and we talk about these theories where you say, okay, you start from simple bars and light pattern, and then you put them together across multiple layers of a neural network in order to reach a face, for example. The experiments, the video that I'm showing you is from Hubel and Wiesel. This is an important set of experiment in neuroscience, and they won a Nobel Prize for it. So these are important stuff, and at the time it was very revolutionary to see, oh, a single cell is doing something so meaningful, right? That kind of was revolutionary at the time. People didn't think that you could find, it's such a complex system. There are so many cells, they're doing so many different things. But for some to come along to show that no, this single cell is doing something interpretable, something meaningful. And a lot of theories that came after that kind of relied on this finding. So using a technique like that, you could really map out, you could put an electrode somewhere, and then you could move it a little bit, you can move it a little bit. And as you saw, even in this little video, you saw that they were doing a lot with that light. They make it bigger, they make it narrower, they have different orientation for it, right? You could, and because the animal is anesthetized, you could close one eye, flashlight in one, and then another, and then just find out how cells are responding based on those factors, based on the shape of the light, based on the position of the light, based on which eye it's coming through. And then that way, you could actually go different part of the visual cortex. Again, this is something, I'm just giving you some teasers of just so you know what we're going to talk about later on. So you could go to different part of the visual cortex and see, okay, which pattern of light in which part of the world coming from which eye activates this area the most. That way you could actually basically assign a function to every little patch of the visual cortex, something like that. And you could call them basically, these are detecting certain pattern of light or something like that. We'll talk about this a lot more later on. But this is possible using this technique. So what are the pros of such an approach? Well, it gives you a very fine-grained idea of what patterns are being detected by different cells. Like, what does this cell care about? Like, this is a very specific localization of function. We're saying, okay, this is specific cell that I put my electrode next to among these billions, tens of billions of cells does this. So that it gives you that to the extent that you could believe it. What are the limitations of this approach? Well, it's really not possible to use it outside animal models, cats, monkeys, or mice, or some of the most used animal models. And it's limited to single cells. Now, both of those things may not be strong limitations in some ways with the changing technology and maybe willingness of people to be implanted. But let's say there are cases where you could actually implant single cells or arrays of cells. Sometimes you could put them all next to each other and implant it. It looks like a little comb. You could just implant the whole thing. What is a case in humans that you could implant electrodes that justifies implanting electrodes? Okay, that's a good one. Sometimes implants. People are willing to put implants inside their ear, or implants in their visual cortex, basically because the pathway from their eye to their visual cortex is not functioning, they're willing to actually have a camera. The camera gets the image, and they directly can implant something in their visual cortex and send the signal, though. We'll talk about something like that later on. But what is the most common case of putting electrodes in human brain? Yes, for Parkinson, for example. That's an example where you place an electrode deep in the brain and give them different shocks. What is another thing that you put electrodes, you keep people in the hospital in order to find something? Okay, yeah. Seizures. If someone has seizures, you want to find what the source of that seizure is. So what people do, you plant electrodes different part of the brain, and they stay in the hospital. They're constantly monitored. And you want to see where the seizure is starting from. It's usually starting from a certain brain area, and the pattern of activity of a seizure is very different from normal functioning brain. It's just flashing, basically. The whole brain is flashing on and off. And you could find the source of that. And then they cut it out, basically. They just cut part of the brain, and then they hope for the best, usually. That's how it works. But if they are in the hospital and they have implant in their brain, then you could collaborate with the scientists. They could come in, they could show them images because they're bored and stuff, or they play video for them, and they can record from certain brain area because all the electrode is already there. That's a case. That's the most common case of researchers getting neurophysiology data from humans. And we'll look at some papers down the line on that. That actually the data was collected that way. It's limited to a single cell. Well, it's not the case anymore, really. Now we have electrodes that could record up to a few hundred cells at once. Like the electrodes are so advanced, and so and the technology has improved so much that from one electrode, you could really record up to a few hundred cells surrounding that electrode. Also, you could put a grid of electrodes, as I said. You could have these grids of 64 electrodes implanted. As I said, it looks like a comb, or yeah, you just implant the whole thing. It's not a single cell. But these are generally the limitations. Even when you have 64, well, it's 64 cells. Or if you have a couple of hundred, it's a couple of hundred cells among tens of billions of cells, right? So it's still a limitation. Even if you could put it in seizure patients, it is still a very limited number of things. So these are still limitations, but there are exceptions to get around. Any questions about this? Well, as I said, we'll watch more of that video because we're going to talk about neurocomputation a little more, and I think that video teaches you something about what different cells are doing. Now, every cell, when I talk about like a cell is coding for this or coding for that, that cell is coding because it's connected to other cells in a certain way, right? Because selectivity, right? If a cell is only selective and responds to certain kind of thing, if a cell is only responds to phase stimuli, well, the only way for that cell to be selective is because it has a certain pattern of connectivity. Connectivity defines its function. In a neural network, connectivity defines function. This is an important point that you should all kind of internalize. That the way that a cell responds is because it's connected in a certain way. It's connected to some other cells that maybe detect nose and hair, and then that nose cell is connected to some other cells that detect, edges and holes and whatnot. So across multiple layers, selectivity, more selectivity, more selectivity, and then a phase cell could emerge maybe many layers down because it's connected to other cells. So connectivity defines function. All right, another neuroscience technique that you may have seen are event-related potentials. These are basically non-invasive. You put electrodes, some of you may have participated in some of these experiments, you basically put electrodes on the scalp, and you inject a lot of gels in there to make it more, to increase the conductivity of it and everything. But you basically measure electrical brain activity from the scalp. But because it's from the scalp, it has to be a lot of neurons. A lot of neurons should be doing kind of the same thing for it to pick it up. Um, and it really builds on the assumption that you have a lot of neurons, as I was saying, a lot of neurons that are active together for the signal to be picked up. So the signal itself is EEG. You may have heard that. It's like electroencephalography. It's an EEG signal. But if you actually have a point where you show an image or you play a sound and you have all these EEG signals, then you could average them out and then get something that we call event-related potential. Because you have an event that happened at time 0, and then something happens maybe 200 milliseconds later, 500 milliseconds later, and you say, okay, this signal shows regret, or this signal shows visual perception. This signal shows that, and this is very common in like um, you know behavioral psychology. People use these markers a lot. But they are usually an average, because the EEG signal itself coming from the scalp is very noisy. So you have to average out a lot of trials in order to get some clean signal. Like as I said, it has to be a lot of cells active together for you to pick up that signal on the scalp. This is event-related potential. And then you could visualize it. You could say, well, which part of the brain is more active, which part of the brain is less active, and you could plot it in this scalp form, and each of these dots is a location of one of these electrodes that is touching the scalp. And you can say, oh, after 200 milliseconds, this area was active. After some 100 milliseconds, that. And you could call these different markers for different type of behavior and whatnot. That's something people use, and this is called scalp topography. Um, so what are the good things about this approach? Well, you could really use it in humans. It's non-invasive. It's fairly easy. It's somewhat affordable, um, and it has very high temporal resolution. That the signal that you're detecting is a signal at that moment. It's not a delayed signal. It's the same for neurophysiology, right? The signal you're detecting is real time. And we're going to talk about other methods where the signal is a little delayed. But in this case, you're recording electrical activity, so the signal is real time. What are the limitations? Where the spatial resolution is really poor. You're not really saying, okay, imagine neurophysiology, right? Okay, spatial resolution in this case means in the brain, right? So this like how, like if you're recording from a whole chunk of the brain, it's not really a spatially high spatial resolution in the brain tissue. That's what the spatial resolution means. This scalp activity is measured, and it's not really able to get deeper in the brain, right? A lot of brain areas are deep, but this is really coming off from the scalp, so it's like really surface. So if you see this, I'm going through these because you're going to see them in the papers that you read, so I want you to have some familiarity with them. Another neuroscience technique, the oldest of them all, was basically brain lesion, right? So this was, a lot of time it happened naturally, right? It was people having a stroke or a tumor somewhere. Sometimes it were accidents, people having concussion, or there is a famous example of someone, a metal bar going through their head and everything, and kind of damaging their prefrontal cortex. It could be a gunshot during a war or something that happened a lot. It could be surgery that you remove a piece of the brain, as I said, because of the seizure or other reasons. Or it could be in animal studies where they basically zap a part of the brain with some electrical activity in order to study what this part were doing. Or it could be reversible. You could actually inject some chemicals to really deactivate a certain area of the brain. And as soon as the chemical kind of wears off, the brain goes back to normal. But the benefit of that is that it's reversible. Now, what is the major assumption here? Well, the major assumption is that if a certain area of the brain is damaged or deactivated, then we observe some change in behavior. If we observe some change in perception, in cognition, it must have meant that that area was somehow involved. You can't say that, well, that area was the locus or location of that percept or cognition, but you could say, well, that area was involved in some ways, which is a very weak, form of connection, but it is, you could say that, well, that area was involved, right? If someone has a prefrontal damage and their judgment becomes shaky after that, or their personality changes in some ways, then you could say, well, this was the prefrontal area is involved in that. That's the major assumption. So a lot of, and as I said, this course is not really going to get too far into anatomy or anything like that. But and as I said, Ellen walked you through these things, but I'm just going to give you some very basics as we talk about this topic of lesion studies. So the brain has four major lobes, right? And we have the frontal lobe, parietal lobe, occipital, which is in the back of the brain. And this is the view of the brain. So it's someone facing that way. And this is the temporal lobe. We talk about the way visual activity travels through the brain in a later slide. And we pay more attention to this, which is closer to the eye, which is not closer to the eye. But for now, just know that there are four lobes in the brain. And people using some of these lesion studies very early on kind of figured out that these different areas may be involved in different things. And these are some of the big ones, right? That okay, the motor control and touch and haptic is like somewhere up here, that the visual perception is somewhere back in the brain, that the language processing is somewhere in the temporal lobe, that some of the, as I was saying, some of the smell stuff is pretty much above the nose or something like that. And these things, and face recognition, for example, a lot of people with damage inside their temporal lobe have problem detecting faces, recognizing faces. So one study that kind of looked at this, and it was an interesting study, was about this concept of visual neglect. So again, this is a view of the brain from the left side, and this is a view of the brain from the other side. So when you see a brain, you kind of have to detect where this folding of the temporal lobe happens, so you could see where the front and where the back is. So this, in this case, like this would be the occipital area, it would be the vision area. So if there's a damage to this part of the brain, if there's a lesion to this part of the brain, people have shown characteristics that have been called visual neglect. And what is visual neglect? Well, people become kind of unaware of certain part of the visual environment. So this is, for example, in this case, if you have a cross and the patient is asked, well, can you reproduce this cross? Well, they kind of don't see what's going on. Now, this is the lesion to the right parietal area or the right parietal area? Which side of the parietal area is lesion here? Right. Why? Who said right?
I did, but I said it because it was on the previous slide. It said.
Okay. But you know that the right, so we talk about this later on. I say that a lot, but we'll do, is that the left visual world is processed by the right hemisphere, right? So the brain flips these things. So this must be a lesion to the right hemisphere in order for you to neglect the left side of the visual world. because it flips the visual input. So when a person with this type of lesion is asked to copy a figure, they are almost unaware of the left side of the visual world. And it kind of, you could see this, like when they're asked to draw that thing, again, they're kind of unaware of the left side of the visual world. And you could see this in a lot of different ways. Not only they're unaware, they are unaware that they are unaware of that the left side of the world, right? They don't know that they're missing something. To them, it looks like a complete figure. So in a way, it's not blindness, right? If you did not see part of the world, but you knew that you're not seeing a part of the world, that's blindness. But if you are not even aware that you're not seeing part of the world, then that's neglect. That's why it's called visual neglect. So this must be damage to the right parietal lobe. Again, coarsely, and I want you to know coarsely that up here is the parietal lobe, occipital, temporal, and frontal. Just coarsely know. So when you see a brain, you kind of have some understanding of where things are. And the eyes, in this case, would be over here. Okay, so the visual neglect really refers to the perceptual suppression of a stimuli appearing in the visual field, opposite of lesion's brain hemisphere, because that's what brain does. Basically flips. It not only flips them horizontally, but also flips them vertically. So later on, we look at maps of where the visual input actually maps onto different brain areas, and the brain flips them in both ways, because that's the property of a lens. That's what how a lens works, right? If you have a lens and you're looking at something, it flips the figure in because the way the light travels. And our eyes just are like a lens, so they kind of flip the image in both ways. Okay, what are the pros of this approach? Well, you could say it gives you somewhat a straightforward connection between a lesion and a brain function. It's debatable whether that's a real thing, that is actually, because you could say, well, this area could be involved in a lot of different things and in some very indirect way be involved in this process, and that's true. But people have taken this to be evidence for these sorts of connections. Well, what is the cons? Well, as I was saying, it's really hard to interpret it. It really is not easy to interpret. Well, if you have a damage to your frontal cortex, which part of it is damaged? You know, it's a whole big field, big area. It's not really possible to say it. And also, with like reversible damages, brain has a way of reconfiguring itself. Right? As I said, there are so many different connections connecting one layer to another layer. If you damage some of them, there's another way for the signal to be communicated. There's so much redundancy, something called degeneracy in the biological systems that makes them robust. That's why somebody could be born with half of their brain and also like have a function normal life, because brain has a way of reconfiguring itself. So it's hard to see, okay, if someone had an injury 5 years ago, you really don't know how much their brain has reconfigured. So it makes it really hard to make interpretation of that. That's for brain lesions. Another thing that people use for experimentation is transcranial magnetic stimulation. This is a form of stimulation that is completely non-invasive. And you may have seen something like this before, but it would be nice. Let me see. Look at this video.
Hi, Recon. Welcome to our laboratory. Today, we're going to show you how the transcranial magnetic stimulator works. So first thing, Yoshi's going to put the magnetic coil over top of the area of my motor cortex that's responsible for the muscles of my hand and forearm. When she's there, she's going to deliver a magnetic pulse. Oh, and you probably saw my arm twitch there. And we can also see this response over here on the oscilloscope because we've measured it using the EMG electrodes over the muscles of my hand and forearm. Now what we're going to do is move that magnetic coil over the span of my motor cortex from one side all the way over to the next. And what you'll see is muscles slowly recruited from my hand up to my elbow, shoulder, right leg. And after she switches hemispheres, we'll see some of the left leg come in and then down the muscles of my left arm. So here we go. So right hand, little bit of elbow, some shoulder and leg, still right leg, little response in my left leg there, we've switched hemispheres, more leg on the left side, left shoulder, left elbow, and left hand. Thanks for joining us.
So what that shows you is that you could really see in a lot of cases, okay, so you could see that people could really stimulate. So you could kind of find a mapping between those areas and maybe certain part of the brain, right? And this would become some sort of a functional specialization. You could say, well, this area must be controlling these muscles. This area must be controlling the muscles of the hand or the shoulder or this leg or that leg. And people go as far as, plotting these body parts on this, and basically look at this. So again, this is the brain. Again, where am I? This is the brain. This is the temporal lobe. So this is the parietal where it meets the frontal. And you could see that the green area is basically sliced to be here, and the red area is sliced. And people have mapped out all these things, saying, okay, the neural activity here is connected to your toes, your knees, hip, and they found all of these, connections, and they call it motor cortex because it moves your body when you stimulate that area. Okay. And also you could basically have a similar thing with somatosensory where you could touch different parts of the body and then see which part would be active, right? You could do this with vision. The video example that I showed you is a vision example. You flash and light different areas, you see what the activity is. You could do that with touch too. You could actually have a sensory input on different parts of the body and see which neurons become more active in this part of the brain. And that way you could kind of map out the connections. Okay, so brain imaging, there are two popular brain imaging methods. You may have heard of them. One of them is PET, positron emission tomography, and the other one is functional magnetic resonance imaging, or fMRI, which is a variation of magnetic resonance imaging, which is MRI. And they really basically measure the brain activity, but not in a direct way. What they actually measure is the hemodynamic response. And the assumption or the finding is that, well, in order for the neurons to communicate, they need energy. All those ion channels that we talked about, they take a lot of energy to function. Pumping out the ions in and out takes a lot of energy. It takes a lot of glucose for them to function. And it means that if a certain brain area or certain patch of the brain is being very active, it would need more glucose. Needing more glucose means that it would be having a higher flow of blood to that area. Now, if you had, and that means, well, it's higher oxygenation of the blood that reaches there. And if you could detect that, then you could detect, well, what's the activity of that area. So it's an indirect way, right? You're not directly measuring the neural activity. You're measuring the blood flow to certain brain areas. And the argument is that, well, the blood flow should be correlating with the neural activity. So what we call the blood flow is basically the hemodynamic response. And I said the assumption in all of this is that the greater activity in a brain region results in greater blood flow to that region. But you could imagine already that because it's an indirect thing, then it's not really very spatially, like you cannot get a single cell level, which is actually measuring activity. But another thing that we'll talk about later, it's a little slower too. For these reasons, we call them indirect. So with PET, we basically you inject and these are humans, right? We inject the human with a substance, and it's a little radioactive substance basically, and it's in their blood system, and when there is activity in the brain, you could basically track this radioactive signal as it moves around in the brain. If there is higher blood flow to certain brain areas, then you should be able to see that there's more radioactive signal coming from that brain area. And the radioactive thing is basically glucose substance, because that's what would be taken up by these brain cells. It has to be a sugar-based thing that could turn into energy. Okay, so basically the overview is that you have a stimulus that makes a firing in the brain, or you have an emotional state that makes a certain firing in the brain. That's gonna need more glucose uptake, and that glucose uptake means that you could basically track this substance, which is glucose based but also radioactive. So when people are in this scanner and injected with this material, then you could track how much blood flow is in different part of their brain. And you could basically see, well, this part is more active, this part is less active, depending on what they're doing. If they're reading something, maybe a certain part would be more active. If they're in a certain emotional state, or if it's in, if you're studying addiction and they're being shown certain figures, or being told certain things, or you could see which part of the brain would be more or less active. It kind of gives you a general overview. So, but you could see something about these methods that is different from the, let's say, single cell neurophysiology. It gives you an idea of the entire brain. What's the entire brain doing, kind of thing. So it gives you a better spatial resolution, and you could visualize. So when I say better spatial resolution, I mean that the activity is not averaged out across many cells and many brain areas. You could say, well, this activity actually belongs to this brain area, or activity belongs to that brain area, something that you couldn't do really with, say, EEG or ERP, because the activity is averaged out spatially. That's what I think people before had a little misunderstanding about what I mean by spatial resolution here. Spatial resolution in the brain, that you could say this patch of the cells are having this activity, or this patch are having that activity. Okay, what are the pros of this approach? As I said, it has relatively good spatial resolution versus some other methods. What are the limitations of such an approach? Well, the temporal resolution, because you're recording the hemodynamic response, you're recording the blood flow, and the blood flow is much smaller than neural activity. The neurons are functioning on a millisecond basis. Blood flow functions on seconds, right? So if an area is taking more blood, you'll see it 2 seconds later. But if a cell is being more active, you see it in a millisecond basis. So imagine how different in time these operations are. But this is just a proxy for it's just a proxy for the brain activity. It's not really brain activity. So it has a poor temporal resolution because the blood flow is small. Blood flow kind of tails the activity, but also because it exposes people to radiation. So you basically are having radioactive material injected into people to see where these activities going to. So it's not really something that you could do too many times. Any questions? These are just a few techniques that I have to go through because you'll see them later in your papers and we'll discuss them. So it's just good for you to be exposed to them.
Are PET scans ever used to diagnose anything, or they just use?
I think they're used for diagnosis too. Some sort of, I know of addiction diagnosis and stuff like that based on PET scans.
They're used really widely in oncology.
Okay.
Yeah, they're because the difference, it picks up on a difference in metabolic rate that can indicate like malignant tumors and things like that.
Yeah.
What would you use if you wanted to look at like entropy?
Entropy.
Yeah,
In what way?
like let's say somebody took psychedelics
Okay.
and now there's entropy, right?
Mm-hmm.
That neural networks are getting created.
Neural networks are getting created, you
I'm
said?
not sure if that's
Okay.
the same to say.
So I think
Curious.
I'm not familiar with the studies, but I'm sure people have looked at brain activity under substance and how it would be different. I'm not familiar with it, but if it changes brain activity to the extent it may be visible in the fMRI stuff that we're going to talk about. I'm not too familiar with those studies under substance, but I'm sure you could look it up and see how is brain activity different under LSD or other substance, and then see how it looks like. I'm not familiar with it. Okay, so fMRI is measuring the blood oxygenation level. It's saying, okay, if there is blood flow to a certain area, this blood is the fresh blood, and it is oxygenated, like the hemoglobin cells carry oxygen. And this signal is basically, as I said, is a slow response. This is not something that you could really measure right away. If there is activity, increased activity in a certain brain area, it will increase the blood flow to that area maybe 3-4 seconds later. So it's a slow response. You're not measuring activity at that moment, and you're not really measuring activity. You're measuring blood flow. So if a region of brain starts to consume about maybe 2%, maybe there is maybe 2% more oxygen in that brain area. So what you do is that, well, you have a condition that they're not looking at anything. You have the brain activity. Then you have another condition that they're looking at an image or seeing a movie or making a judgment. Then you subtract the two. And then you say, okay, this activity must be related to this stimulus, right? When you do an experiment, you want to know what is the impact of this stimulus versus that stimulus. So you want to have a baseline. And this is how you set up a baseline. In your design, you have places where there is nothing, and then you subtract it. Let's get through this so we can be done with it. We're going to start something new next time. So what is the benefit of this? Well, fMRI has kind of the best of both worlds in some ways. It is limited in some ways, but it gives you an image of the entire brain. It gives you, well, and you could overlay it on the MRI, which is the structural. It gives you the structure of the skull and the brain areas, but then you could put fMRI on top of that and visualize it really nicely. So it gives you some idea of what's happening in the entirety of the brain. But also it gives you somewhat much better resolution than otherwise. Like people have like 7 Tesla fMRIs, right? It gives you up to millimeter by millimeter of the brain. It's still, there are many neurons in that little chunk, tens of thousands of neurons, but you get enough resolution that you could make maybe argument that this is within a certain brain area. So it has good spatial localization. Uh-huh. Not usually fMRI. It's usually the EEG stuff that is used for that. The stuff that we talked about earlier there. Because it's easy. So because in the so fMRI machine is big and expensive.
Oh, yeah, it's expensive and they have to take.
No. fMRI doesn't have any radioactive stuff. That was PET.
Okay.
But fMRI is functional. So the way fMRI would be done, you would have to wheel them into a machine, and then they have to be stable there, which, as you said, if they're in a coma, that's not a problem. But usually the what happens is that they are usually in a people that like that are in, let's say, a coma state. They usually have EEG on their brain. So the stuff that we talked about earlier. They have some electrodes on their brain, and those can detect markers of some conscious behavior or not. It's not an easy thing, but that's yeah. So fMRI gives you precise spatial localization, and it's non-invasive, and you don't inject anyone with a radioactive anything. So it's a great thing, right? It's not invasive. It gives you good spatial resolution, but it has a very poor temporal resolution. That when you have something happening, you can only detect it a few seconds later. And also, it's a technologically demanding thing, and it's expensive, right? It's a big deal when an institution has fMRI machine, so people can study it because it's an expensive thing. Let me show what I meant. So when you ask that question, I think what's more common for the patient in the hospital to have, it's something like this. So they have some electrodes attached to their brain, and that's what they monitor. And it's activity of a big chunk of the brain on basically below the scalp. All right, that's it for today. I'll see you all next week.
I reserved this bag of food, but I need to go pick up before 8, so I need to run.
What's that? Or let me see.